Welcome to the Deep Dive, your shortcut to cutting through the noise and really getting well informed. Today, we're taking a fascinating plunge into the world of Mpox, formerly known as monkeypox. We're not just skimming the surface here, we're going microscopic, exploring its, well, surprising origins, how scientists classified its really intricate structure, and the clever ways it gets around inside our cells. Our source material for this is a great overview, drawing from Mpox, history, classification, structure, and cell entry by Elena Monat, which also pulls in key insights from the CDC. Now, you might think you know Mpox, but did you know its actual origins are still like a bit of a mystery, even with that name? Okay, let's unpack this.
That's right. And our mission today isn't just to list facts. We want to help you understand the why behind them. From the very first time this virus was spotted, right down to its complex internal bits and pieces and how it interacts with your body at a cellular level. Think of it as peeling back the layers, really getting the full picture.
Okay, so let's start with the history, which is genuinely surprising. Mpox was first identified way back in 1958, and not out in the wild, but during 2 outbreaks in colonies of research monkeys. In Copenhagen, Denmark, actually, hence, the original name, monkeypox.
Exactly.
But here's where it gets tricky. Even though it showed up in monkeys first, scientists still aren't totally sure where it really comes from naturally. That name, monkeypox, it's kind of misleading, isn't it?
It is a bit. The current thinking, the suspicion, is that African rodents and maybe some other non-human primates are the natural reservoirs. They might carry the virus and then sometimes pass it to people. And while it was found in those labs in '58, the first actual human case wasn't recorded until 1970. That was in the Democratic Republic of Congo.
Right. And then if we jump forward quite a bit to 2022, we saw this huge shift in its global presence. Before then, cases outside the areas where it's usually found or it's endemic were super rare, almost always linked to like international travel or maybe imported animals. But then, yeah, it just seemed to spread globally. Quite suddenly.
It really was a dramatic change. According to the CDC data, looking back as of July 30th, 2025, there had been about 36,000 cases worldwide, and 5 cases specifically noted in the USA by that point. What's really fascinating here, I think, is how something once seen as geographically limited can just suddenly become a global issue. It really brings up an important question. What is it about this virus that gives it the ability to have such a widespread impact?
That's a perfect lead-in, actually, because understanding that global potential often starts with knowing its family, its place in the viral world. And this is where it gets really interesting, connecting Mpox to some other well-known, maybe even feared viral relatives.
Right. So when scientists classify it, it's taxonomy. Mpox belongs to the family Poxviridae. Within that family, it's in the genus Orthopoxvirus, and the species is Orthopoxvirus monkeypox.
Okay, orthopox virus.
Yeah, and that puts it in the same group as some really significant viruses from history. Smallpox, for instance, also called variola, and also vaccinia virus, which was used in the smallpox vaccine, and cowpox virus too. They're all related.
So why does that connection matter for us? What's the big deal about it being related to smallpox?
Well, that's actually a crucial point. Knowing it's an orthopox virus is a huge advantage for researchers. It means it shares fundamental characteristics, maybe even vulnerabilities, with viruses like smallpox that we've studied for, well, centuries. This isn't just academic trivia, It gives scientists a massive head start when they're thinking about treatments or vaccines. They can build on all that existing knowledge from its cousins. It's why, for example, the smallpox vaccine offers some cross-protection against Mpox. That relationship is key.
Got it. Okay, now let's shrink ourselves down, metaphorically speaking, and look at how this virus is actually built. It sounds like it's quite a unique structure.
It really is. So Mpox is what we call a linear double-stranded DNA virus. That just means its genetic blueprint is made of DNA, like ours, but it's a single straight piece, not a circle. And its genome, its instruction manual, is pretty big for a virus, somewhere between 170 and 250 kilobases. That codes for about 200 different proteins, so quite complex.
Thanks. Linear dsDNA.
And here's something really unique about its genome. It has what are called covalently closed hairpin ends. Imagine taking a piece of string and tying a sealed loop at both ends. Most DNA has free ends, like loose shoelace tips. Mpox DNA is basically sealed shut. It gives it extra stability, helps protect the genetic code, and probably plays a role in how it replicates once it's inside a cell.
Okay, and its shape and size, you mentioned it's large.
Yeah, absolutely distinctive. It's often described as brick-shaped, roughly 250 nanometers long and 200 nanometers wide. It's genuinely one of the largest viruses known, huge in the virus world.
Wow.
And another key thing, Unlike lots of viruses that head for the cell's nucleus, Mpox is a cytoplasmic virus. It basically sets up shop and does its work out in the main body of the cell, the cytoplasm, not in the central control room, the nucleus, which is unusual for a DNA virus.
Right, and it has an envelope, an outer layer.
It does, yes. It's an enveloped virus. That means it has this fatty outer membrane, which it actually steals from the host cell it infects as it leaves. If you could see the whole virus particle, the virion, you'd notice it has roughly 5 main parts. There's the core, a membrane around that, these things called lateral bodies, surface tubules on the outside, and the nucleocapsid holding the DNA.
Okay, quite a few components.
Yeah, the core itself is sort of dumbbell-shaped, which is interesting, and it's wrapped in a double-layered membrane. And on the very outside surface, you have these tiny little spikes or protrusions about 10 nanometers long called surface tubules.
Okay, and inside it carries its own tools.
Exactly. Crucially, Mpox virions pack a whole bunch of enzymes related to making RNA from DNA, the RNA polymerase system. Think of them as the virus's own little toolkit or mobile factory inside its shell. These are vital because they let the virus start making copies of its genes and building new virus parts almost immediately after it gets into your cell. It doesn't have to rely entirely on the cell's machinery for that first step.
Very efficient. And the genome itself, the DNA, you said it has different regions.
That's right. It's about 197 kilobases long, this linear molecule. And you can broadly divide it into a large central region, which is pretty stable and conserved across related viruses.
Right.
And then 2 variable regions at either end, the terminals. And right at the very ends of the DNA are these structures called inverted terminal repeats, or ITRs. They're complex bits containing gene sequences, hairpin loops, like we mentioned, and short repeating sections.
So what do these different parts do?
Functionally, that big central region holds the instructions for the essential stuff, the structural proteins that build the virus particle and the core enzymes it needs just to function as a virus, the basics. But those variable regions at the ends, the left and right terminals, that's where the really interesting stuff often is in terms of infection. They contain most of the genes that determine which hosts the virus can infect its host range and how severe the disease is its virulence genes. Think of them as the virus's adaptability toolkit.
Wow.
And connecting this back, understanding these precise details, the structure, the genome layout, it's absolutely critical for scientists trying to design antiviral drugs or vaccines. It helps them pinpoint specific targets. Like knowing the blueprints lets you find a weak spot or a crucial part you can block. It's much better than just guesswork.
Definitely. Okay, so we have a good picture of this brick-shaped invader. But how does this large, complex thing actually get into our cells? And then how does it spread?
Yeah.
This part of its life cycle, from the cell's point of view, sounds really fascinating.
Yeah, it is. So Mpox actually exists in 2 main infectious forms. There's the intracellular mature virion, or MV, that's the main form found inside the infected cell. Then there's the extracellular enveloped virion, or EV. This one has an extramembrane layer and is specialized for moving between cells and spreading the infection further.
2 different forms for different jobs.
Exactly. Now, how it gets in? Mpox enters host cells, basically, by attaching to the surface and then fusing its membrane with the cell's membrane. For getting out, the MVs are typically released when the whole infected cell just breaks apart. That's called lysis. But the EVs, they exit more subtly, using a process called exocytosis, where the cell actively pushes them out in a little membrane bubble.
And it's still kind of a mystery, isn't it? Even with all this, the main key or receptor that Mpox uses to unlock our cells hasn't been definitively identified.
That's right. It's one of the big unknowns still. A bit frustrating for researchers, But we do know some things. We know Mpox can bind to molecules called glycosaminoglycans, GAGs, which are these long sugar chains on the cell surface. That seems to help it dock onto the cell.
Okay, so it uses those GAGs.
Yes, as attachment factors. And because it's an enveloped virus, there's also a theory, a postulation, that it might use something called apoptotic mimicry.
Apoptotic mimicry. What's that?
It basically means the virus might trick the host cell into thinking it's cellular debris or a dying cell fragment. Cells have mechanisms to clean up that kind of stuff by engulfing it, so the virus might exploit that cleanup process to get swallowed.
Wow, that's sneaky.
It is. So once it's attached, either via a specific receptor we haven't found yet, or using GAGs, or maybe this mimicry trick, it fuses its membrane with the cell's membrane. This fusion event basically opens a doorway, allowing the virus core with its DNA and enzymes to be released directly into the cell's cytoplasm.
And once inside, it actually relies on some of our cell's own machinery to help it, right? These complex names, COG and JAPAR.
Yes, exactly. It turns out the virus hijacks, or at least heavily relies on, two multi-part cellular machines called tethering complexes. One is the conserved oligomeric Golgi complex, or COG complex. The other is the Golgi-associated retrograde protein complex, or GARP complex. These are normally involved in managing traffic inside the cell, particularly around the Golgi apparatus, which is like the cell's sorting and packaging center.
So what do they do for the virus?
Well, the COC complex helps regulate things like adding sugars to proteins, called glycosylation, and moving membranes around. The GARP complex, working with proteins called VPS52 and VPS54, helps maintain the Golgi's structure and function. And what researchers found is that these complexes, especially GARP and those VPS proteins, are absolutely essential for the virus to form those EV particles, the ones that spread infection efficiently and generally for a successful Mpox infection cycle.
Huh. So the virus needs our cell's internal postal service and decoration department to work properly.
That's a good way to put it. It highlights how interconnected the virus is with the host cell's normal processes. And this immediately raises an important question for research. Why are these specific cellular components so vital? If we can understand that dependency better.
Maybe we could block those pathways and stop the virus.
Exactly. Understanding these interactions could be crucial for developing new ways to interrupt the infection process, maybe targeting the host factors the virus needs rather than the virus itself.
Wow, what an incredible journey we've taken today. We've really dug deep into Mpox. From its, well, surprising start in those Danish research monkeys and the ongoing mystery of its true origin, all the way to its intricate brick-like structure, those unique sealed DNA ends, and the really clever ways it seems to navigate getting into and out of our cells. You've just gained, hopefully, a pretty thorough understanding of a complex topic quickly and effectively, going way beyond the headlines. Hopefully you feel better equipped now to think about Mpox with a bit more depth than maybe you had before.
And maybe a final thought to leave you with. Given everything we've discussed, that unique structure, the unknown origin, that still missing host cell receptor, what else might we uncover about how this really fascinating complex virus interacts with our cells? And what could finding that receptor, for example, mean for future ways to prevent or treat it? Could that be the key breakthrough?
An excellent question to ponder. And a great reminder that in science, especially virology, there's always more to learn, always another layer to peel back. Until next time on The Deep Dive, keep that curiosity going.